Pollen is the male gamete in flowering plants, and it can be a useful way to store plant genetic resources. Although pollen is almost never used independently to grow an entire plant, it is an essential resource for breeding and conservation programs, particularly when the desired plants are at different locations or bloom asynchronously. Because of its value to plant breeders and those who conserve rare species, pollen has an important place in preservation programs, and luckily it can sometimes be easier to preserve than either seeds or clonal tissues such as shoot tips. Desiccation-tolerant pollen is usually released from dehissing anthers with moisture contents of 30% or less. Pollen can spread by wind or insects to receptive flowers, and then seeds are produced. This desiccation-tolerant pollen can likely be cryopreserved. Some self-fertile plants, such as wheat, release pollen that has moisture contents of greater than 50% and does not tolerate air drying. Therefore, desiccation-sensitive pollen is more difficult to process and store. Scientists at the National Laboratory for Genetic Resources Preservation in Fort Collins, Colorado, conduct pollen preservation research as well as process and store pollen in liquid nitrogen. This material is gathered from living plant collections across the country that are maintained by the USDA National Plant Germplasm System. Since research is still needed to establish reliable methods for more sensitive pollen, our facility currently preserves only those that are desiccation tolerant, such as pollen from pecan, date palm, and prunus trees. Here's an overview of the process used to store pollen at the NLGRP using prunus pollen as an example. Flowers are harvested either just before the pollen matures or as it is beginning to be shed. Enough should be collected to meet conservation goals. Pollen must be processed quickly, preferably within 3 days of harvest. This includes drying the flowers or catkins, sifting to separate the pollen from the floral organs, then shipping overnight to NLGRP. Upon arrival, the pollen is equilibrated in conditions that provide about 50% relative humidity, usually over saturated salt slurries of calcium nitrate at room temperature. The next morning, the pollen is ready to be distributed into storage vials. The vials are then immediately placed into storage in the vapor phase of liquid nitrogen, where the pollen remains viable for years. Our current goal at NLGRP is to preserve a minimum of 15 vials for each gene bank accession. The size of a vial and volume of pollen is determined by the practices used to pollinate each crop. For instance, prunus pollen is preserved in half milliliter aliquots, while date palm pollen, which is used in greater volume, is stored in 4 milliliter vials. Viability is ideally tested by making sure trees can be successfully pollinated. However, logistical difficulties such as access and timing make this type of assessment impractical for large-scale processing. Instead, we use an in vitro germination assay and measure pollen germination levels on medium. To do this assay, a vial of pollen is removed from cryostorage and a small amount is placed in a 98 to 100% humidity environment for 4 hours. It's then plated onto growth medium containing calcium nitrate, sucrose, and other nutrients and placed in the dark at room temperature overnight. The next day, pollen tube lengths are measured using a microscope. A pollen grain is considered germinated if the length of the pollen tube exceeds the diameter of the grain. All inventory and germination data are recorded in our online database, Grain Global. Research is underway to improve and expand pollen cryopreservation methods. One of the biggest challenges for successful processing is the need to minimize the time from harvest to liquid nitrogen storage, which can be tricky when plants are growing in remote locations. For a new crop, it usually takes a season to determine best practices for pollen collection, processing, and viability assessments before the pollen can be routinely stored. While continued research is needed to better understand the longevity and use of stored pollen, one thing is for sure: pollen provides conservation programs with the opportunity to preserve male gametes to help secure the future of plant genetic diversity.